we are talking about enzymes. And first thing first, you need to know what an enzyme is. And I believe by now what an enzyme is because it's been it's been a discussion from high school. So an enzyme, we define it as a biological catalyst. Catalysts are just things that can make the reaction to move faster. Let's say, for example, I want to convert A into B. What is going to make A to be converted into B much faster? That is a catalyst. But an enzyme is not just another catalyst, it's a biological one. Biological. And every time you're talking about biology or biological, it is living, okay? Or doing its work in living organisms. So biological catalyst. Now, another thing that you need to know in the definition of a catalyst is that it will make a reaction move faster, but it is not changed. That is very important. It is not changed. So the reaction is moving much faster, but what is making the reaction move faster is not changing. What makes up enzymes? That should be another question that we should be able to answer for us to understand enzymes. So enzymes are going to be made up of proteins. Okay, proteins. Now, I want us to be reminded when we looked at proteins, we said that proteins have got levels of protein structure. For example, what makes up proteins are amino acids. So if you get this amino acid, connect it to this amino acid, you connect to this amino acid, another amino acid, just like that. We call this as a primary structure. Primary structure is only connected by what? Peptide bonds. So the primary structure will only show peptide bonds because the bond between one amino acid and another amino acid is a peptide bond. Now, this primary structure that we have formed can start now folding about itself. So it can fold in a spiral way. How do I even show a spiral way? So it can fold like that. Okay, you can see the way it has folded. That's a bit spiral. So this is known as, or let me first show the other one. Another way it can fold is known as a beta pleated. So it's going to fold something like that. So this structure on top is known as an alpha helix. And the one down is known as a beta pleated. So these two structures are secondary structures. Okay, secondary structures. Now, of these secondary structures, a secondary structure, what we are doing is that we are getting a chain, a long chain like this, a long chain, and then now it starts to fold about itself. It starts to coil like that. Take note that the secondary structure is only formed from one chain. Now, the secondary structures can further coil around about themselves to form tertiary structures. And what you form is either a globular shape or a fibrous shape. So how does globular look like? Globular, the proteins are folded like something like that. They make like a bow. And then the fibrous, you form more like a fiber. Okay, more like a fiber like that. So this is a tertiary structure. When you get more than one tertiary structure, you put them together. You form now a quaternary structure. So for example, hemoglobin is a quaternary structure and is made up of 4 subunits. And you can see it's globular. And then you also have collagen. Collagen is also a quaternary structure because it's going to have 3 chains put together to form it. But in an exam, the most important structure for collagen is a secondary structure. They do ask that question. But it also can form a tertiary structure Tertiary structures, you get more than one polypeptide that has folded into a quaternary structure is a structure where you get more than one polypeptide folded into the tertiary structure. Enzymes are globular in nature. Okay, so this is how they look. They are globular. And it is important for you to understand, because we are saying that they are globular. What bonds holds globular proteins together? Peptide bonds hold the primary structures. Secondary structures will have peptide bonds, including hydrogen bonds. Now, tertiary structures will have peptide bonds, hydrogen bonds. You also have what we call ionic bonds. You are going to have disulfide bonds. You are going to have hydrophobic interactions. You are going to have a lot of different bonds you can see in the tertiary structure. And the quaternary structures are made up of tertiary structures. So meaning that enzymes which are globular in nature, they are going to have all these bonds: ionic, disulfides, hydrophobic, hydrogen, the peptide bonds. Please take note of this. That's very, very important for you to know. Okay. So there are a lot of things that can affect the way enzymes will carry out their enzyme catalyzed reactions. And the first thing that you need to understand is the concept of active site and specificity. So we need to know that enzymes are specific, meaning that each enzyme, just like we have got a law. So here one of the theories we use is the lock and key. But just know that enzymes are specific. So when you look at the structure of an enzyme, it's going to have two sites. So imagine this is our enzyme here. So for this enzyme, it's going to have the part where the other compound that has to be converted is going to bind to. So remember, you want to convert A into B. Let's say A looks like this, okay, and then B is this. How do you do that? What you do is that you are going to get A, you fit it here. This point where A is going to fit is known as the active site. Active site. And this A that you want to convert into B after fitting in the active site is known as a substrate. Substrate. And what you are getting after you convert is known as a product. Okay, like that. Now, apart from that, take note of the shape of the enzyme. It is globular in nature. Enzyme have got another site where the substrate will not bind, and that is known as the allosteric site. So there are some things that can bind to the allosteric site, and when they bind to the allosteric site, what is going to happen is that they can either make the enzyme to fit well with the substrate, or they can make the enzyme not to fit with the substrate. Okay, please take note of that. So what is the work of the active site? The active site is there to bind our substrate. How about the allosteric site? Allosteric site is the entire actually the remaining part that is not the active site, and that is going to be used. It's the remaining part of the enzyme that is used to maintain the correct globular structure. So if you get to play with the allosteric site, you are going to disturb the globular structure of the enzyme. That is very important for us to know. Okay. What other thing are we supposed to understand here? Another thing that you need to understand about enzymes is that they are specific. And that has to do basically with the kind of active site that the enzyme you are dealing with has. So for example, you can see this enzyme that is here, it has got active site that is a bit rectangular. So the substrate that is going to fit in is going to be like this. So at the beginning we had our substrate. The substrate reacted with the enzyme. What did we form? We have formed this structure here. What is the name of this structure? This is known as the enzyme substrate complex because it has got both the enzyme and the substrate. This enzyme substrate complex is going to break. What are you going to see at the end of it all? The enzyme will not change its shape, it will be the same way it's looking. But then the product is gone, the substrate has changed. So at the end of the reaction, you still have the enzyme, but then you don't have the substrate. The substrate is now the product. That is very important, okay? That is very important. So about specificity, what we are saying is that each enzyme is going to have only one substrate it can work on. One enzyme is equal to one substrate. That's what we mean if we say enzymes are specific. So, who proposed this theory or this model? This is known as the lock and key model. The lock and key model, the other name is also known as the Fisher model. So this model was developed by the scientist Fisher who proposed it, and this was in the year 1890. And according to Fisher, he suggested that an enzyme is going to have a site that is going to exactly fit with the substrate. So the substrate is like this, and you can see the way the enzyme is. And when the enzyme fits with a substrate, you are going to first form the enzyme substrate complex before you can form the product. So you are going to have enzyme plus substrate, then you have got enzyme substrate complex, and then after that, then you are going to have the product. Okay, that is it. Now, another scientist came up and said, actually, the enzyme is not fully doesn't have a very complementary shape as that of the substrate. So what did he say? He said that enzymes will actually get to change their shape a bit so that they can accommodate the substrate. And that is known as the induced fit theory. Induced fit theory. Okay. Induced fit theory. And the induced fit theory is also known as the Koshland by the name of the one who proposed the Koshland model or theory. So this one, what are we saying? It was proposed by who? It was proposed by Koshland. In which year? 1958. So according to the induced fit theory, let's say for example our enzyme is like this, and then our substrate is like this. So according to this model, the enzyme is going to change its shape a bit so that it can fit with the substrate. So the enzyme gains a shape like this. And then now the substrate can come and bind there to be able to fit. You can see how it has changed. So that is the induced fit theory or model. That is what you need to take note about this theory. So there are different factors which are going to affect enzyme catalyzed reaction, and we need to know them. The first thing that you need to know is that enzymes are biological catalysts. Okay, biological catalyst. Now the question is, how does a biological catalyst work? Biological catalysts, they work by lowering the activation energy barrier. Please take note that it's lowering. And these are the same exam. These are exam questions. Trust me, if you don't want to know it now, you'll be forced to start wanting to know it in October. You need the data very much. Okay? Biological catalyst will lower the activation energy. I can simply write like this: activation energy barrier. And how is that? So here is a graph. This side is showing us the energy that is needed for the reaction to happen. This side is showing us how the reaction is progressing. So some enzymes will go like this. So here you have got your reactants. Before you can have your products, you need to give the reactants enough energy. And when you reach, it's like you need first, before you can form the product, you need first to climb the mountain. Just like before you can get a degree, you have first to go through the studies. You can see the studies are bit pressuring, isn't it? So you need to endure that pressure, and that pressure makes you to spend a lot of energy. You're spending a lot of energy on books, spending a lot of energy on everything else. And when you reach the top part here, then you get your degree. Okay, like that. Here now you have got your product. So the distance from where the reactant is to the maximum point is what we call activation energy. Here I wrote EA, but normally we will write, I wrote AE, we'll write it like that. This is activation energy. Now you also have the difference in energy between the reactant and the product. Here, this difference. This is known as the enthalpy, but that's not even our interest, or the overall energy released during the reaction. So meaning that for this reaction, it's supposed to be endothermic because the energy of the reactants is reducing. Okay, we understand that in chemistry. So what is an enzyme going to come and do? The enzyme will come and lower the activation energy barrier. Instead of you climbing a very long mountain, you can see the mountain has become shorter. Because as long as you climb the mountain, then you are going to reach the product. So this is the reaction with the enzyme. The one on top is without the enzyme. So you can say the activation energy has been lowered, isn't it? So that is the aim of an enzyme. Very, very important. Okay. Another concept that you need to understand is the concept of cofactors. Remember, we said that enzymes are going to have an active site and an allosteric site. So, what are cofactors? Cofactors are things that an enzyme requires for it to be able to bind to the substrate. Actually, enzymes will not function without cofactors. They will not function. Okay, they need cofactors. So, some enzymes require things which are non-protein. Cofactors are not proteins for them to become active. So, cofactors can either be, we can divide them into organic or inorganic, and we need to know the difference. Normally, when you're talking about something being organic, it has got carbon hydrogen bonds. That's the meaning of organic. So, cofactors which are organic, these are also known as coenzymes. So, a coenzyme is not anything special, it is just a cofactor that is organic. While cofactors which are inorganic, these are just going to be metal ions. So, you need to know examples. Okay. So when an enzyme that requires, let's first talk about. So you have got this enzyme. This enzyme cannot work without a cofactor. So when an enzyme that requires a cofactor is not bound to its cofactor, it is known as an apo enzyme. Check through last year's paper and the 2021's paper. You will see this. So what is an apo enzyme? An apo enzyme is an enzyme that requires a cofactor, but it is not bounded to its cofactor. But if you get this apo enzyme plus its cofactor, okay? So apo enzyme, so apo enzyme plus cofactor there. Apo enzyme plus cofactor, this forms what we call an holo enzyme. It's very easy, okay? A plus C is equal to H. Hapoenzyme plus coenzyme is equal to holoenzyme. In 2022's exam, they were just changing this. Holoenzyme plus cofactor equal to hapoenzyme, different options given like that. So when the hapoenzyme is bounded to its cofactor, it's known as an holoenzyme. And this is the active form. Please take note that the hapoenzyme is inactive. This is active. Okay. I've said that enzymes don't work if they are not bounded to their cofactors. So cofactors, how do they bond to their apoenzymes to form holoenzymes? They do it covalently. They form covalent bonds. That's also important, covalently. But even if it's a covalent bond, they are not very tight. They are not very tight. Okay. So basically that is it. So what examples are we able to give on organic cofactors and inorganic or coenzymes and the metal ions which will act as cofactors? So coenzymes will include the vitamins, and this one also 2022's 2021 exam, you can check it, you'll find it. Mobita, do you have a question? Okay, so if there is no question, please don't open your mic, or if there is no contribution. So coenzymes include vitamins. Apart from vitamins, you have also riboflavin. If you are coming in medicine, you also find me. I'll get to take you in courses in medicine also. So riboflavin and also thiamine, also folic acid. So vitamin B12, vitamin B9. Okay. So all these things that you're able to see here are going to be coenzymes. They are not the only ones. You also have NADPH, ATP. There is also what we call coenzyme A, also known as CoA. We don't finish it as coenzyme, we just say CoA. So all these here, if you find them, they are all organic cofactors, also known as coenzymes. How about inorganic? They are just metal ions like magnesium ion, zinc ion. All these are going to be cofactors. Okay, just that. Now, let's talk about the rate of enzyme activity. So there are different things that will affect the rate at which enzymes will be able to work, or factors that affect the rate of an enzyme catalyzed reaction. The first factor is temperature. So we need to be able to understand how does temperature affect the rate of enzyme catalyzed reaction. So what you need to know about temperature is, say we have got a reaction diagram here. This side we are talking of the rate of the enzyme catalyzed reaction. This side is showing temperature. When the temperature is very low, enzymes will be slow. But as you get to increase the temperature, you can see the temperature is increasing, the rate is also increasing. and you are going to reach the maximum point where enzymes will be able to work very, very well. And this is known as the optimum temperature. Optimum. And then beyond optimum temperature, the rate of the reaction starts to decrease. What does this mean? In this region, the reaction is slow because enzymes are inactive. Okay, the kinetic energy is little for the enzymes to work. And then in this region here, the reaction is slow not because of being inactive, but because the enzyme has been denatured. We have destroyed the enzyme. That is why the reaction is slow. And when remember, an enzyme is actually globular in nature. So what have we destroyed? You don't destroy primary structure. Remember what we started with? What we've destroyed is actually the secondary structure and the tertiary structure, okay, of the enzyme. So we have disrupted the hydrogen bonds, the ionic bond, the disulfide bonds. You don't destroy peptide bonds in denaturing. Okay, that is all we need to know for now. For those enzymes which work in our bodies, they work up to an optimum temperature of 37 degrees. Like that's the normal temperature for our bodies, between like 36.5, 37.5. So that is the temperature at which they also work, following the environment in which they are working. Our bodies normal temperature is 37. So even the enzymes in our bodies normally work at 37. If you increase that, they will be destroyed. Another factor that is going to affect the the enzyme catalyzed reaction is pH, potential hydrogen, pH. So pH also, it is going to be the same. Below the optimum pH, the enzyme catalyzed reaction is slow, and beyond the optimum pH, the enzyme catalyzed reaction also is slow. So this is the optimum pH. So here you have got the reaction getting to happen, the rate of reaction, and then here you have got pH. You can see as you get to increase the pH, you reach the maximum where the reaction is going to be very much faster, and as you get to exceed that, the reaction is going to become slower. Okay, so what do we need to understand here? The enzyme will become denatured and the rate of the activity is going to be reduced. So changes in pH leads to changes in the ionic charge of the acid or the base. Remember, we are saying enzymes are proteins. Proteins are made up of amino acids, and amino acids can either be acidic or basic. So if this amino acid is basic, and then you make the environment to be acidic, you are destroying it. Why? Because what are you really changing? When you change pH, you learn this next year if you're in medicine or any other program in biochemistry that has to do with Ridgeway. Changing the pH is going to change the ionic charge. So you have changed the ionic charge of the enzyme. That is why the enzyme is being destroyed. Changing the temperature wasn't changing the ionic charge. What was it changing? It was denaturing. Okay, it was destroying the bonds. Take note of this, please. So, if you have got an enzyme that is, for example, let's talk about the stomach. You know, in the stomach, we have got what we call hydrochloric acid. Now, sometimes the stomach can produce too much hydrochloric acid, so you can give some medicine like omeprazole. You know people with ulcers in the stomach, right? Hydrochloric acid will start chewing the stomach, and so that leads to ulcers. So if you want to fight that, you are going to give omeprazole. Omeprazole does reduce the amount of acid being formed. So the enzymes which are found in the stomach are going to work well in acidic environment. For example, you have got enzymes that break down proteins, pepsins. You have got enzymes that will break down carbohydrates, that the maltase. So these ones will work well where? In acidic environment. Why? Because the stomach is acidic. When these enzymes move from the stomach, because here is your esophagus, here is your stomach. After the stomach, you're going to have the duodenum. When the enzymes move from the stomach here, they come in the duodenum. The duodenum is basic. These enzymes will be inactive, they'll be destroyed because they've gone in an environment where they cannot work. So take note of that. Okay. Another thing that will affect the rate of the reaction is the concentration of substrate. Now, when you're talking about substrate concentration, how does this affect our reaction? So, here we are going to have the rate, and then here we are going to have the substrate concentration. Let's say, for example, you have got 20 enzymes, and then you have got only 10 substrates. All the substrates which will fit on the enzymes. So when you start increasing the number of substrates to 15 to 20, the reaction is going to be increasing. But there'll be a point when, even when you increase the number of substrates, the reaction can no longer increase. Let's say now you have got 30 substrates. The reaction will not increase, and this is called the maximum velocity or the maximum rate, Vmax. Why? Because increasing the rate of the reaction, increasing the substrate will no longer affect the rate. So you can see the rate will still be the same. Why? Because the extra number of substrates don't have any enzymes that are going to work on them for the reaction to be faster. This is also going to be the same if I change this. I change and I now start talking about the enzyme concentration. Enzyme concentration also affect the rate of the reaction and will also change the x-axis. It no longer be substrate, but it will now be enzyme. Okay. So when you increase the number of enzymes, let's say you had 20 substrates. When you increase the number of enzymes, let's say you had first 10 enzymes, you had 20 substrates. When you increase the number of enzymes, the rate of the reaction will become faster. But when you reach 20 enzymes, that will be the fastest. You are going to have Vmax. If you have now 30 enzymes, increasing the number of enzymes will not change the rate of the reaction. Why? Because you don't have any more substrates for the enzymes to work. So meaning that 10 extra enzymes will not be doing anything because there are no substrates. So that is it on this one. Is there any question? Is there any question? Be free to ask if you have got any question. Okay, let me talk about inhibition and then we answer some exam questions on this topic. So we have talked about things that can speed up the rate of the chemical reaction, but then there are also things that will inhibit or hinder the way enzymes are going to function, and we call that as inhibition. The things which inhibit, we call them inhibitors. Okay. So a number of substances can reduce the rate of enzyme catalyzed reactions. Now, when we are talking about those things that inhibit enzyme catalyzed reaction, what do we call them? We call them inhibitors. Things that will reduce the rate at which enzymes work. Now, these inhibitors can either be, you can either reverse their function, reversible, or they can be non-reversible. Okay. So that is one important thing you should take note. They can be reversible or non-reversible. What do we mean? Inhibitors that are reversible. Let's say, for example, here is our enzyme. And then this inhibitor came and bound here. It changes the structure of the enzyme. And then it makes the active site to become like this. So initially, the substrate that can work on this enzyme can work on is supposed to look like this. But if the shape is like this, you can see that the enzyme can no longer work, isn't it? So that's why inhibitors can be reversible, non-reversible. Okay. Those sometimes when you increase the number of substrate, that is going to overpower the inhibitors. And so the way the inhibitors were inhibiting the reaction, they will no longer be able to inhibit the reaction. So you have reversed it. The enzyme is now working normally. But non-reversible is where if the inhibitor just binds, there is nothing that you can do. The enzyme is destroyed like that. It can no longer speed up the reaction. That is the non-reversible. Now, these inhibitors again, we can as well say they can be competitive or non-competitive. So what is going to be the difference? So for competitive inhibitors, they bind to the same site where the substrate is supposed to bind. And the site where the where the substrates bind is the active site. So where will the competitive inhibitors bind? They'll bind to the active site. So when look at this, this is the enzyme. This is the active site. This is where the substrate was supposed to bind. And then you have got this inhibitor, it comes and binds there. What to happen? The actual substrate which was supposed to bind there cannot bind there, and that slows the rate of the enzyme catalyzed reaction. But when you are talking about non-competitive inhibitor, these ones will be able to bind to another site. And that site where they will bind will not be the active site. These ones will bind to the allosteric site. But how do they inhibit the reaction? Let's say the enzyme was supposed to be like this, and then this inhibitor came and bound to the allosteric site, and then the active site changes. You can see because we have changed the active site, the enzyme can no longer work the way it was supposed to work. So the way non-competitive inhibitors do is that they change the conformation of the what? Of the active site by binding to the allosteric site. And so the substrate is okay, the active site is there, but then the substrate can no longer fit in the active site. So these inhibitions, the competitive can either be reversible or non-reversible. Same with the non-competitive. It can either be reversible or non-reversible. So reversible means mostly for the reversible, you reverse when you increase the number of substrates. Okay, you have the inhibitor. Okay, I've got one inhibitor. I bring in 10 substrates so that they overpower the inhibitor. That's how you can reverse these inhibitions. Okay, just that. So basically, that is it on enzymes. Let's go to exam questions. Before I go to exam questions, please, the exam questions we are going to have done the part of teaching. So the answering is you who is going to be answering. Are we good? Is it okay that you be answering? If you're not correct, and then I tell you that you're not correct. Is that okay? Be free to open your mic and answer me. The allosteric site. Okay, so this portion here is the active site. The rest of the part that has remained, which is not the active site, is the allosteric site. So what inhibitors do is that when they bind to the allosteric site, that is going to cause a change to the active site. You can see it was somewhat pointed, but it has now become rounded. So because you have changed the binding of the inhibitor changes the active site, that makes the enzyme not to be able to speed up the reaction of the substrate is also supposed to speed up. Is that okay? Okay, all right. Okay, so let's go. This is the first question. In the stomach, proteins are hydrolyzed or broken down by the enzyme known as number one, trypsin, whose optimum pH is alkaline, lipase, whose optimum pH is acidic, pepsin, whose optimum pH is acidic, amylase, whose optimum pH is alkaline, maltase, whose optimum pH is acidic. Which one would you pick? Okay, it's pepsin. And remember, we said that the stomach is acidic, so optimum pH is going to be also acidic. So this is the answer. Take note that when you're naming enzymes, they end in ASE, except some special enzymes which end in IN. You can see like PACE, maltase, amylase. But if they are not ending in ASE, they'll end in IN, like pepsin, trypsin. Okay, so the answer here is 3. Let's go to this one. Choose the statement which is true concern about enzymes. Enzymes do not require cofactors. Okay, you'll be telling me if it is true or false, each option. Enzymes do not require cofactors. Is that true? This one is false. And then two, enzymes do not affect the amount of activation energy required by the reaction. Is that true? We have got a lot of people. Why is it that only one person is answering? The person who is answering, I can assure you, is the only one who is learning. You might think you are learning. When this question comes, you'll be surprised if you answer it. Okay. So is two true? This one is not true. Okay. Okay. All right. So when you answer, you can switch off your mic, especially if your room is noisy, and then you open it when answering again. Enzymes do not affect the amount of activation energy. They do affect. I just explained that if you have got a reaction like this, enzymes do. Mhm. Okay, okay. All right, thank you. So enzymes do affect the amount of activation energy. Okay, good. So my reading was wrong. That's why I've seen people are picking it. Okay, this is true. Enzymes increase the amount of activation energy. Is that true? Okay, enzymes lower, isn't it? So this one is not true. And then for enzymes decrease the amount of activation energy required by the reaction. Is that true? Okay. Vedred, why have you said no? Let's hear your. Mm-hmm. Why? Yes, because. Okay, the because has ended. I've seen a hand. Cecilia. Okay, you can. Cecilia, you can come in. Mm-hmm. 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 Okay. So look at this. If this is a reaction, The energy from the reactant, here is the reactant, here is the product, to the maximum point. This is what we call as activation energy. What an enzyme is going to do is, because the reaction can't happen until you climb this mountain from where I've written R to the top. So enzymes will lower the energy that you take for you to climb this mountain like this. So enzymes will lower the activation energy. Is that okay? Okay, and okay, yes, there is a hand. Please, you can go on. Mm-hmm. Yes. Okay. Okay. So this is why when you're answering biology questions, if you pick one answer as correct, don't forget about the other options. You say, I've already found the correct answer. No, you don't do that. I would have just said, okay, two is correct. Let's go to the next question. But I didn't do that. That's why I want us to look at all the question options, because you just realize that this answer two is not necessarily wrong, but it's not the best option. Okay, so that's why you have to check through the entire options, because you just find the best. Here we only want the best. Remember, there's negative marking in biology, so you can actually get negative negative 100 if you are not careful. Okay, so option three, enzymes. You can get negative 100 instead of getting 100%. So option four, enzymes decrease the amount of activation energy. This is correct. Option two is not wrong, but it can, as our friend has said, it can either be positive or negative. That's why it's not the best options. Is this okay? Is this question good? Okay, let's go to this one. The lock and key theory of enzyme was proposed by Fisher, Koshland, Chagaff, Watson and Crick, Rosalind Franklin. Mm-hmm. Which one is the answer? Yes. Okay, by Fisher. Cecilia, what did you want to say? Mm-hmm. Same answer. All right, very good. And then Koshland, what theory did he propose? What theory did Koshland propose? We've already forgotten. Good. Someone to help him finish. Induced what? The induced fit. So it's induced fit. You don't always fit in, but you want to induce, like you want to change so that you fit in. We'll talk about these other guys here in the next topic. We'll talk about to be nucleic acids. We'll also do it the same. We just like we have done all the topics below. Do the topic answer exam questions, so that you don't just know the topic without answering questions. So we'll talk about those guys in genetics and nucleic acids. The optimum pH of the enzyme trypsin, which is found in the small intestine, is acidic above pH 7, neutral, either alkaline or acidic below pH 6. I do not know. Which one is the answer? Which one can you pick? Above pH 7? Okay. Vedred says acidic. Anyone else? Cecilia? Above 7? Anyone else? Acidic. Anyone else? Okay, now I want to get reasons. Those who have said above 7, And those who have said acidic, can you tell me the reason for your answer? Okay, thank you. And then those who have picked acidic, let's have one. Yes, Cecilia. Okay, thank you. And then those who said acidic, Vedred, you've changed your minds. Tell us if you've changed your minds. Uh-huh. So please, this is what happens. Sometimes we students we just swatter questions. You swatter a question, but you swatter the wrong question. Okay, so that's pretty. Okay. So remember, as I explained, I said that the stomach is acidic and pepsin will work well there. When you go into the small intestine, like the duodenum is where our friend was explaining some gallbladder. Are you coming to Ridgeway next year? I think it will be interesting with you talking about gallbladders now. So if we have got people who be doing health sciences, medicine, pharmacy, nursing, oh, you'll find us next year. Okay, so what will happen is that in this region, the small intestines are basic. So any enzyme that is acidic coming from the stomach will be neutralized and it will stop to function in the small intestine. So the answer is above 7. It is basic. It's actually 7.5. A- Any molecule which acts directly on an enzyme to lower its catalytic activity is called coenzyme, cofactor, inhibitor, substrate, a product. Which one would you pick? Yes, Cecilia, an inhibitor. Anyone else? Do we all go for inhibitor? Okay, inhibitor. Okay, very good. The next one, which of the following is a coenzyme? Is a coenzyme. Metal ion, protein, vitamin, purine, lipid. Which one would you pick? Vitamin. Anyone else with a different option? Vitamin. Is there a different option? Hmm. Only two people. Let me get from two other people. Data ya soba. Okay, so the answer is vitamin. That's correct. The optimum temperature for most human enzymes is 25, 30, 35, 37, 100. Which one would you pick? 37. You go for what? 37. Okay, Cecilia. 37. Okay, very good. In the induced fit model of enzyme activity, number one, substrate changes its shape slightly after binding the enzyme. Number two, enzyme active site changes its shape slightly after binding the substrate. Number 3, both enzyme and substrate change their shape after binding each other. Number 4, none of the enzyme and substrate change their shape after binding each other. Number 5, the enzyme and the substrate have a perfect fit of each other. Which one is the best option? Okay, so Hussein, is that your name? Hussein. Oh, you're using someone's email. Okay, very good. So Hussein says to anyone else, he says that the enzyme active site is going to change its shape slightly after the binding of the substrate. hmm So I only know Hussein, Cecilia, and Vedred because they're the ones who only one speaking, and Movita. Uh-huh. So the others who I don't know the names. What would you say? Movita says number two also. Cecilia, what would you pick? Vedred. Ah, you don't. Okay, so I think. Okay, so you people didn't attend when I was talking about the induced fit. You joined a bit late, or maybe you didn't understand. Okay, I see. You should follow up the recorded session. I've talked about anything and everything that can come in the exam. The effect of a competitive inhibitor on enzyme activity is that the effect of competitive inhibitor, it reversibly increases enzyme activity, irreversibly stops enzyme activity, irreversibly increase enzyme activity, reversibly change the shape of the enzyme active site, reversibly stop enzyme activity. Which one would you pick? Let me start with option one. Reversibly increase activity. Does it increase the activity? No, so this is not correct. How about two? Irreversibly stop enzyme activity. Does it irreversibly stop? Okay. Uh, okay. Which one do you pick? Or what do you say about option two, Movita? Okay. And then option three, irreversibly increases enzyme activity. Is that true? It's not true. And then reversibly changes the shape of the active site. Is that true? Okay. Since you've said no, I've put an X. And then reversibly stops enzyme activity. Is that true? Okay, no. You say. Yes, Cecilia. Okay. So let me clarify. When you have got an enzyme, when you have got an enzyme The inhibitor comes to bind to the allosteric site. Allosteric site. The shape of the active site is going to end up changing. Here we're talking about a competitive inhibitor. Competitive inhibitors don't bind to the allosteric site. They bind to the active site. So that's why option 4 is not correct. Okay? Reversibly changes the shape of the active site. What to change? Competitive inhibitors will bind to the active site and stop the binding of the substrate. What binds to the allosteric site and that changes the active site are what? Are non-competitive inhibitors. Okay. And then you also, another thing, competitive inhibitors, when they bind to the active site, when you increase the amount of substrate, you can actually overcome the competitive inhibitor. It's like you're competing for the same girl. So the one who has got more money, if she loves money, is going to win. So when you increase the number of substrates, they'll win over the competitive inhibitor. So competitive inhibition is actually reversible. So reversibly stop enzyme activity. This is a correct answer. Option 2 is not correct. Irreversible. It means that even if you increase the substrate, you'll not stop you'll not what? You'll not help with the reaction. No. So the best option is 5. Have I made it clear? Okay, normally those which are going to be irreversible can be non-competitive. As I explained earlier, the non-competitive can either be reversible or irreversible. So I've got some which are reversible, some are irreversible, including also on the active side, because some things will come and bind to the active site, but the only thing is that they bind there permanently. That makes them irreversible. Anything that to form permanent covalent bonds is irreversible. But remember, I want the best option. So the best option is 5. That is because most of those things to come into the active site, you can overcome them if you increase the substrate. Because before this guy, the inhibitor comes to bind here, if you have got a lot of substrate, it will go there and prevent the substrate from binding there. Otherwise, if it forms a strong covalent bonds, it binds there, the process becomes irreversible. Does that make sense? Yes, Cecilia. Okay. A poison, the only thing that you should know, poisons are organophosphates. The only thing that you should know for now is that it is nearly irreversible. Nearly irreversible. But it will not act on enzymes. Of course, there are kind of enzyme. It acts on what we call receptors. So imagine this is a receptor. You have got what we call now I'm teaching you medicine. So there are things in our bodies where neurotransmitters. Neurotransmitters are things which control our nervous impulses. So when this neurotransmitter comes and binds to this receptor, then it makes you maybe to know that you are actually under pressure or there is a lion so that you can be able to react. But then organophosphates, these poisons, they will go and attach there and that will stop the binding of the actual neurotransmitter. And so everything that is supposed to be done is reduced. And that nearly takes it takes, they will tell you 100 days 100 hours for it to move out, and by the time it's moving out you're already dead. Okay, so that's why it's nearly irreversible. Okay, is there any other question before we go on? Okay, let's look at this one. A non-competitive or an allosteric inhibitor, A, changes the shape of the substrate. B, binds to the substrate. C or 3, binds to the enzyme active site, competes with the substrate, does not bind to the enzyme active site. Which one is the best option here? 5. Okay. Any different option or anyone to support? No one? 5. Okay. Okay. Okay, it does not bind to the active site. But I can see the screen. Maybe try to check your bundle. You might be out of bundles. Okay. So the best option is 5. So if it does not bind to the active site, the rest of the enzyme that's not active site is a allosteric site. Let's look at this one. Choose the statement or statements which explain why enzymes are good biological catalysts. They are consumed in the reaction they catalyze. They are very specific and their effect is reversible. They lower the activation energy for the reactions they catalyze. They are rich in nucleotides. 2 and 3 are correct. Which one would you pick? Option 5, 2 and 3. Okay. Anyone else? Option 5. Okay. So why is option 5 correct? You tell me. Mm-hmm. Mm-hmm. Okay. Okay. So when enzyme binds to substrate, you form the enzyme-substrate complex, and then you form your enzyme plus product. These reactions can also go in the backward direction, so that's irreversibility. Okay, so we can pick option 5. Which of the following statements regarding enzymes is false? Enzymes are proteins that function as catalysts. Enzymes are specific to their substrate. Enzymes lower the activation energy for reaction. Enzymes acti- activity cannot be inhibited. Enzymes may be used many times for a specific reaction. Which one amongst this is not correct? We want what is not correct. Let's start with option 1. Is it correct or wrong? Okay, 1 is correct. How about 2? Enzymes are specific. Is also correct. How about 3? 3 is also correct. How about 4? Okay, 4 is not correct. You know that enzymes can be inhibited, right? And then 5, enzymes may be used many times for the same reaction they catalyze. For this, for a specific reaction. Yes, you can use them for a specific reaction many times as long as the substrate is there. The temporal association between an enzyme and a substrate can best be described as, remember, it's temporal association between enzyme and substrate. 1, an enzyme substrate complex. 2, a covalent bond association. 3, one in which the enzyme is changed permanently. 4, a permanent mutual alteration of structure. 5, complementary base pairing. Which one is correct amongst these? Yes, Cecilia. Option 1. Okay. Tisa, do you want to pick an option also? Option 1. Okay, good. Okay, that's the answer. Okay, let's look at this one. The active site of an enzyme always remains rigid and does not change, is found on the surface of a globular protein, is the biggest part of the protein molecule, contains amino acids with the same side chain, is also called an inhibitor site. Which one would you pick amongst these? Mhm. Let's start with an option. Option 1 is always remains rigid and does not change shape. Is that correct? Remember the induced fit, we said that it can change. How about option 2? Is found on the surface of a globular protein. Is that correct? Friends, we said it's like this. Look at this. Look at this. You can't see that it's on the surface of this is a globular protein. Okay, let me leave out that option. Is the biggest part of the protein molecule. Is that correct? The biggest portion is the allosteric site. Contains amino acids with the same side chain. Is that correct? It's made up of different amino acids. So different amino acids have got different uh, side side chains. It is also called an inhibitor site. Is that true? So you see what the answer is? Okay, option 2. It's found on the surface of the globular protein. That's the surface. Which of the following bonds can be found in enzyme molecules? Ah, this one if you fail. Ionic bonds, are they found in enzymes? Someone has said no. Peptide bonds, are they found? Someone has said yes. Disulfide bond bridges, are they found? Hydrogen bonds, are they found? So what's the answer? Someone, we already ticked one row out. Okay, so everything is correct. So remember we said that primary structures of enzymes are made up of peptides, and then secondary structures are going to have hydrogen bond. But for you to have a secondary structure, it means that you have a primary. Definitely secondary also has peptides. And then tertiary structure is going to have peptide, hydrogen, disulfide bonds, hydrogen bonds, and also ionic bonds, hydrophobic bonds. Now, if you get more than one tertiary structure, that forms a quaternary structure. For example, hemoglobin, myoglobin, collagen. Okay, so the answer is all of the above. Which of the following statements describes how the substrate fits with the enzyme? The lock and key hypothesis. The active site changes shape slightly after binding the substrate. The induced fit hypothesis, the substrate fits exactly into the enzyme active site, all of the above. Which one is the answer? Which one is the answer? Hmm? 2. The active site changes shape slightly. Okay. Anyone else? Where are the people we are shining? That red. You've stopped shining. Cecilia. Okay. The answer is all the above. Look at this. Option 2, the active site changes slightly. That is describing option 3, which is the induced hypothesis. And then option 4, the substitute fits exactly. That is describing option 1, which is the lock and key. So it's all the above. Do you get the sense? Hope mukayimona mu exam. Okay, now next one. Which of the following compounds permanently reduces enzyme activity? Remember, permanently. Competitive inhibitor, reaction products, irreversible inhibitor, cofactor, coenzyme. Which one would you pick? Mm, it's just direct. It's just direct, huh? It's irreversible inhibitor. Okay, so it's option three. Okay, this is where my questions ended. Our drive contains other questions. And if you get to access our drive, we have got past papers. It should be like from, I think it's 2012 up to 2023 exam papers. You can revise and revise and revise. The goodness with biology is the constantly repeat, not biology. NS, the constantly repeat, repeat, and repeat. And we have got solved questions, past papers in all the courses.